Chapter 7 is all about cellular respiration, and this is basically how cells make energy. Okay, there's a variety of ways that energy is made, and there are various forms of cellular respiration. And one of the things that we are going to take a look at in the beginning of this chapter is aerobic respiration, which is going to be a type of cellular respiration which requires oxygen. Okay. Before we get too far into it, though, we do need to review this molecule, which ATP, which stands for adenosine triphosphate. So if you look at the molecule, it's something like this. This whole part here is called adenosine. It's made up of one ribose sugar and a nitrogenous base called adenine. That's the same one that is found in DNA. Okay, and then it's called TP triphosphate because there is 1, 2, 3 of these phosphate groups on the molecule. And right in this bond right here, this is a high energy bond, okay? And that means that it takes a lot to put that phosphate on to the ADP. By the way, if you just have one of these phosphates, it's called inorganic phosphate, PI. And then if you add that to this molecule that only has 2 phosphates on it, we call that adenosine diphosphate. So if you add a phosphate onto adenosine diphosphate, this is what equals ATP. Okay, so this is going to be mostly what this chapter is about, how this is done. Okay, there's kind of a pretty clever technique that the cells will employ to make the ATP by putting a PI onto ADP. But in either case, that energy that's stored in this bond can be broken anywhere in the cell to release that energy. So the phosphate can be added to ADP. It can then travel to somewhere in the cell, and then this bond can then be broken, and then all of the energy is released to drive some sort of biological process. So keep that in mind. This is ATP is a big champion in this chapter. Okay. All right. So here's how it works. The whole overview for cellular respiration, or at least aerobic cellular respiration, looks something like this. You take a molecule of glucose. The glucose, remember, is C6H12O6. You add a little oxygen into the mix, and what you get out is carbon dioxide, water, and 36 ATP. Okay, and if you do the math on this, it should all balance out, right? So over on this side, you could, if this was, let's say, the reactants, and let's say this side is the products, and we're looking at how many carbons, oxygens, and hydrogens are over here, and how many carbons, oxygens, and hydrogens are over here. Here we've got 6 carbons, and we've got 6 oxygens here, but then we also have 6 molecules of 2 atoms of oxygen bound together, so that's 12 oxygens here. This is 6 oxygens here. 12 plus 6 would be 18 oxygens, and then we have 12 hydrogens. Okay. Well, over on the other side, then we've got 6 carbons, and then we've got 6 water. Water is H2O, remember? So 6 times H2 would be 6 times 2 is 12. And then the oxygens we've got, again, are 12 oxygens here because there's 6 molecules of CO2, and each molecule of CO2 has got 2 atoms of oxygen. So 6 times 2 is 12, plus 6 times 1 oxygen atom per water molecule, so that equals 18 as well. So here's kind of what happens, right? You have this carbons and the oxygens and glucose, they get converted into carbon dioxide and also water, okay? And then the hydrogens are going to be utilized to make ATP, but then they end up forming some water as well, okay? So all that to say, what happens to all of the sugar that you eat in the morning and all of the glucose that's found in all of the different foods that you eat throughout the day, ultimately one of the fates of it is to be stripped down, and then you breathe it out as CO2, and it accumulates as water, and in the process you make a whole bunch of ATP, which is energy to drive stuff. Okay? This is an important equation. You're going to want to be able to write this down and have this kind of on the forefront of your mind, because hint, hint, it might show up again at some point in time. All right, so keep that in mind throughout this whole process. All right, now that whole thing that I showed you, the conversion of glucose into water, carbon dioxide, and ATP, generally there's 3 steps that we talk about in this process. Okay, so here they are. There's glycolysis, the citric acid cycle, and oxidative phosphorylation. And we're going to spend a good chunk of time here talking about each of these. So for starters, we said we start with a molecule of glucose. Okay, this is glucose, again, is a monosaccharide. It's found in all sorts of foods, and it's one that your body uses a lot. Okay, it's got 6 carbons, 12 hydrogens, 6 oxygens. Okay, well, what happens first is that that molecule actually gets broken down. So this is a 6 carbon molecule here. It gets broken down into 2 3 carbon molecules. You can see 1, 2, 3, and then there's another 3 here for a total of 6. So we're going from glucose to 2 molecules of pyruvate. This is what is happening in this first part called glycolysis. All right. Then what happens is that the pyruvates get modified and converted into these molecules called acetyl-CoA. This little step here is not kind of considered one of the main 3 steps here, but it is an important step, and it's called the oxidation of pyruvate, which I'll tell you more about what that means in a minute. Okay, and then lastly, all of these things are basically acetyl-CoA is going to be totally stripped down to all, so it doesn't have any atoms left anymore, and this is going to be mostly what happens in the citric acid cycle. So that's this step. And then the actual process by which ATP is made, this kind of is referred to as oxidative phosphorylation. Now, why it's called phosphorylation is this. Phosphorylation means to add a phosphate group onto something. And so that's exactly what's happening when you make ATP. You're taking adenosine diphosphate, okay? You're adding on a phosphate, which if the phosphate's just kind of in solution, we call it inorganic phosphate. So that's why we put this little I here. Well, when you add that phosphate to make ATP, that is what's called phosphorylation, okay? Now, why we call it oxidative phosphorylation, this gets us to 2 important terms that we've got to kind of sort out straight away here. Oxidation is one of them, and oxidation is kind of the reverse of what's known as reduction, and vice versa. What oxidation means is to lose electrons. So when a molecule loses electrons, we say it was oxidized. When a molecule gains electrons, we say it was reduced. So when you look at this word, oxidative phosphorylation, it's going to basically tell you that this is a process by which ATP is made because the phosphate group is added to ADP. That's where we get phosphorylation. And then the thing that makes that happen is the loss of electrons at some point in time. Okay, so you take some electrons off of something and you use it to phosphorylate ADP to make ATP. One of the ways that I usually remind myself here the difference between oxidation and reduction, and this is something I still use today if I forget. I always have remembered this little mnemonic. Leo the lion gurs. Okay, Leo gurs. This means loss of electrons equals oxidation, gain in electrons equals reduction. Or if you prefer, you can do this one, oil rig, which basically stands for oxidation is loss, reduction is gain. Okay, either one of those, but just so you want to keep in mind what oxidation and reduction are, because there's going to be a lot of electrons being transferred, and oftentimes what will happen is that one molecule will be oxidized, meaning it's losing electrons. Those electrons will be passed to a different molecule, and then it would be gaining them, so then it would be reduced. So oftentimes one thing is oxidized, and then immediately after it's used to reduce something else. So sometimes we call these types of reactions redox reactions because it's reduction coupled with oxidation. Okay. Anyways, so this is the process. You take glucose, you break it down to 2-pyruvate, you break it down further into the acetyl-CoA, and then use it to make ATP. Now, what I want to start with here is actually the final step, because this is one of the more complex steps. And so it kind of helps to just kind of jump right into that complexity, and then hopefully once that's understood, then maybe the first two steps will make a little bit more sense. So for starters, here's kind of my question, all right? And here's one of the things that I want you to keep in mind. This is kind of the summary of what's happening in aerobic respiration. And I want you to, I've kind of tallied up all of the atoms here so you can keep an eye on them. So when we go from glucose to pyruvate, you can see that we've lost 12 hydrogens, or sorry, 6 hydrogens, right? Because if you add up all of the atoms here and all of the atoms here, that's C6H6O6, whereas all of the atoms here in this molecule was C6H12O6. So we've lost 6 hydrogens. And then from pyruvate to acetyl-CoA, you can see we've lost both some carbon, and we've also lost a little bit of oxygen. And then from acetyl-CoA, all the remaining carbons and all the remaining hydrogens and all the remaining oxygens are stripped, so that by the end of the day we've made some ATP. Well, ATP doesn't have any of those original carbons, hydrogens, or oxygens that were once found in the molecule of glucose that we started with. So my question to you then is, what happens to all these things, right? You start with 6 atoms of carbon, 12 atoms of hydrogen, 6 atoms of oxygen. Well, where do they go? What happens to them? It's kind of important to keep that in mind. You want to know where all this stuff is going, okay? Because that will kind of help you navigate this whole aerobic respiration complexity here. So I'll give you a little hint. The carbon and oxygen atoms, so these ones that you find in the glucose, it's pretty easy. They're going to go straight out to CO2, and there might be a little bit of water that's made from some of those oxygens and hydrogens as well. But basically, all the glucose that you consume, where does most of it go? You breathe it out, right? CO2 is the thing that you exhale out of your lungs. So all of that carbon is kind of being breathed out little by little as you exhale. Now the hydrogens then, this is a little bit more of an interesting story here. All of these hydrogens basically get split into electrons and protons. Okay, recall that a hydrogen atom is going to be 1 proton and 1 electron. That if you add these things together, it equals 1 hydrogen atom. Okay, and so if you have 12 hydrogen atoms, that equals 12 electrons and 12 protons. So protons are sometimes also referred to as this, H+, because if you take a hydrogen atom and you strip the 1 electron from it, you're only left with a proton, but sometimes it's written this way. So keep that in mind, that these are the exact same thing. And then sometimes this is referred to as a proton, sometimes this is referred to as a hydrogen ion. So keep that in mind. Okay. Well, these electrons and these protons, these are going to be the things that are used to make ATP. And so now we will discuss how that happens. And that is the process known as oxidative phosphorylation. Okay. This is a process that occurs in the mitochondria. This is also something that makes a lot of ATP, and as we said earlier, it requires hydrogen ions, okay, and also hydrogen electrons. And it also requires this protein, which is called an ATP synthase. And the name here of this protein should kind of give away what it does. Synthesizing something means to build or make, and if it's an ATP builder, it actually is going to be the thing that makes ATP. Okay, it's the thing that actually puts the phosphate, it's the thing that actually does this. It takes 1 phosphate, adds it to adenosine diphosphate to make ATP. All right? Okay, so here's where we are. All right, we're talking oxidative phosphorylation, and we're inside the mitochondria. Now, I quick want to point out some of the features of a mitochondria. There's 2 membranes to a mitochondria. There's the outer membrane, and there is the inner membrane. There's a pretty important region of space here called the intermembrane space. So that's this kind of the space that you see in between the inner and the outer, or the inner and the outer membrane. Okay, so this is the intermembrane space. And then lastly, there is a part of the mitochondria, that's the very innermost part here, and this is called the matrix of the mitochondria. Okay, so you want to keep these different parts in mind because that will help you remember where we are when I show you some kind of interesting figures here in a minute. Okay, well, all along the inner membrane of the mitochondria, you can find these ATP synthase proteins. Okay, and so their main job, it's pretty simple. They make ATP. So how it works, though, the thing that drives them or fuels them is that hydrogen ions will flow through the ATP synthase, and when they do, the energy of those ions flowing through here is going to take an inorganic phosphate and add it to an ADP, and when you do that, voila, you make ATP. Okay, so on the surface this seems very simple, but the thing that's going to get complicated is to supply these hydrogen ions here in the first place. Okay, to make this whole pump go, there has to be what's called a proton gradient. So these little things, these are protons, and this is in the inner membrane space, right? So this would be the outside membrane of the mitochondria right here, where you've got, you know, the phospholipid bilayer of the outer layer of the mitochondria. So in between the outer layer and the inner layer, this one here, there's an accumulation of protons, and there's a proton gradient, meaning that there's a high concentration of protons, and in the matrix of the mitochondria there is a low concentration. Well, if you recall from a previous chapter, hydrogen or any type of substance wants to diffuse from a high to a low concentration gradient. If there is a difference in concentrations from one place to the next, and there's a high concentration of something one place and a low concentration of something in a different place, and a gradient exists between those two places, the particles are going to go move from that place of high concentration to low concentration. That is just one of the principles of diffusion, okay? And we certainly see it here. Now, so these are all going to want to move from high to low, but they're not able to pass through the plasma membrane because they've got, you know, they're charged particles, and charged particles don't really jive too well with the hydrophobic nature of the plasma membrane. Okay, so they are able to go through, but they require a carrier protein, and that's this one, which is the ATP synthase. And then recall that this would be an example of facilitated diffusion because it's a carrier protein is being utilized to allow that diffusion to pass through. Well, all of this energy that's stored in here is now flowing through, and it's providing the energy to put these two molecules together. It's what's actually making this whole protein, the ATP synthase, work. Okay, so the way I kind of think about it, it's almost like you know, like you kind of think about like those old school water wheels that were like back in the olden day where they're next to like a river, and as the water flows through it turns the wheel, and it was sometimes like connected up to a mill that, like, as the wheel spinned along the water, it would like drive like some sort of pole that like mashed corn into flour or something. I mean, this goes back to like 200 years ago. This is how they did stuff. Well, that's kind of what this thing is. You can almost think of hydrogen ions as being kind of like a river that flows through this pump, and then it provides the energy required. And there, you're going to see in a minute, I'll show you an animation, there's actually something that rotates in this carrier protein as the hydrogen ions flow through it, and that's going to make ATP. So here we are. Take a look here at the ATP synthase.
Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
All right, so this begs an important question, all right? We just saw that, oops. Let me just pause this for a second. I want to just point out something. We started with a high concentration of proton ions. So each one of the, or hydrogen ions, each one of these little things are protons, okay? And they come from hydrogen. You take a hydrogen, you strip the electrons off of it, you're left with a proton. So here they are. And you started out with a high concentration of them. Then, as they flowed through, they made this pump turn, which was the thing that supplied the energy required to make ATP, which is what was happening down here. But over time, all of this stuff that's up here, this is a high concentration, this is a low concentration, and as these are flowing through, eventually the concentration on the outside here and the inside here are going to equilibrate. So then there exists kind of a problem. How do we keep this gradient, the high concentration to low concentration, established? Because as soon as there's an equal number of protons here as there are here, then there isn't going to be a tendency for these ones to flow through, and then ATP will not be made. So there's a couple of questions here that I have for you. The first is, where do the protons come from? And then the next question would be, what establishes the proton gradient? Okay, these are two important questions. Well, all of these protons that you see here, where they ultimately came from is glucose, right? Glucose is C6H12O6, okay? And we said that these were actually broken down into the electrons and protons. So that's 12 electrons and 12 protons. Well, these 12 protons, that's what's making all these things, all right? So when you have a lot of sugar molecules being broken down, here's where all the protons from that hydrogen end up. But still we have to account for the electrons. And the electrons is what is going to be responsible for the answer to the next question here. What establishes this proton gradient? How do we get all of these things, these hydrogen ions that have just flown through, how do we get them back up there? And how do we, store a whole bunch of protons up here to reestablish that gradient so that this will then keep flowing down? And the answer is this special thing called the electron transport chain. The electron transport chain is something that utilizes electrons to push protons from a low concentration to a high concentration. Okay, so here's what it looks like. Just to orient yourself, this is the intermembrane space, so there would be an outer membrane right here. The very outer membrane of the mitochondria would be somewhere like around here. Okay, and there's a series of complexes. There's 4 complexes, and then plus the ATP synthase, which is not shown here. Now, you certainly don't have to know what this looks like. You don't have to know what these complexes are called. That's something that you would learn about more in a biochemistry class. But I do want to point out where we are, and I do want to point out that all of the hydrogen ions are being pushed into that inner membrane space. So this is the thing that's establishing that proton gradient. Okay, And just so you're kind of aware of where we are, this is the outer membrane, this is the inner membrane. So on that same membrane where all the ATP synthase proteins are, that's also where you see these electron transport chains, is what they're called. All right, so I want you at this point to pay very close attention to one particular molecule, and that's this one, NADH. NADH, it seems like kind of an obscure name for a molecule. It stands for something, I forget what it is. It's like nicotinamide something, something or something. But you don't need to know that. All you need to know is that this is an electron shuttle. So when you see this thing here, just know that this is carrying with it 2 electrons. Okay? How it works is there's this molecule called NAD+, okay? And so 1 electron is going to be attached to here to make this just straight up NAD with no plus. And then also a hydrogen is going to then be added to it. Well, hydrogen has 1 electron and 1 proton, right? So then you're adding a second electron onto there as well. So every one of these is going to have 2 electrons. By the way, if I'm adding electrons onto NAD+, what would that be, oxidation or reduction? The answer is that NAD+ would be being reduced, okay? So here's the story, all right? Here's what's going on. NADH is carrying with it electrons, and specifically 2 electrons. It dumps those 2 electrons off at the electron transport chain, which is all this stuff that you see here. The electrons will flow through the series of all of these complexes, and as they do that, there's a whole bunch of hydrogen ions in here, inside the matrix, and the reason these hydrogen ions are here in the first place is because they are there from the breakdown of glucose. Okay, so when glucose is being broken down, specifically this is the citric acid cycle, when that acetyl-CoA is being stripped of all of its electrons and protons and everything, you've got a whole accumulation of protons here, all right? And as these electrons pass through the electron transport chain, the positive charged protons are kind of tricked by the electrons. They see the negative charge kind of whizzing by, and they want to meet up with it. And there's certain places where they're able to get really close, and that's these complexes that you see here. But the way that these complexes are arranged, they allow the hydrogen ions to get enticed or attracted by the electrons, but they don't actually attach to the electrons. So they actually get tricked into accumulating in the intermembrane space. And so before no time at all, the cell has been able to actually move a whole bunch of protons from the matrix of the mitochondria, where there was a whole bunch of them accumulating because of glucose being broken down in the citric acid cycle. And it's actually able to shove all of those or push all of those protons up. And so this is kind of an active transport because you're actually moving these up their concentration gradient because they're starting to now accumulate. Okay, so the thing that's driving that is these electrons that have just come whizzing by. Now, you might ask, well, where does this come from? The electron shuttle. And that is also going to come from glucose, okay? So you had the hydrogen ions that were right here. These came from glucose. And then also the electrons that are being delivered also came from glucose. So that's kind of ultimately the purpose of this glucose molecule, is really to supply electrons and protons that can make this whole electron transport chain work. Now, once the electron transport chain has worked and you've accumulated a whole bunch of hydrogen ions in the intermembrane space of the mitochondria, okay, so here they are all accumulating. Now we have a proton gradient, and then they can basically just flow through that ATP synthase like we talked about, and that's going to make ATP. And then as they flow through, they're going to kind of rejoin this pool of hydrogen ions, which is then going to be pumped back out and then they flow back down through, and then they get pumped back out again, and then they flow back down through, and the process just kind of repeats itself to make lots of ATP. Now, a couple other things I want to point out here. You also see this molecule called FAD, and this is also an electron shuttle, so this can carry 2 electrons as well. Generally, this is the one that's more often used, but this one can certainly be used as well. So they're both electron shuttles. They're basically both just bringing electrons to the electron transport chain. Now, another thing is that it's important to figure out or follow what happens to these electrons. So I already said that they flow through the electron transport chain, but then what happens is that they exit the electron transport chain, and they are actually going to land on oxygen. So oxygen is considered to be the electron acceptor, or the final electron acceptor, because generally, you don't want electrons to just fly through and then be rattling around in the body. That's a bad thing, because electrons can kind of mess stuff up, right? They can break apart molecules and things like that. So luckily, there's a whole bunch of oxygen here that's actually willing to grab onto those electrons and take them up. And what it does is it takes 2 electrons, it takes 2 protons, and voila, forms water. Okay, so if you take some, and it's kind of written in a weird way, but basically what happens is electrons and hydrogens get added to oxygen to make water. So another question I have is, where is all this oxygen coming from? And the answer is, you're breathing it in right now, okay? So the reason you have to breathe right now, as you're listening to me explain about you breathing, is that the oxygen that you breathe goes into your lungs, it diffuses into your bloodstream, your blood cells bring it to all of the cells in your body, it diffuses into the cells, it makes its way into the matrix of the mitochondria to right smack dab here, okay? And then that oxygen its purpose is to be there to take up these electrons that are coming off of the electron transport chain. So that's why you breathe, okay? And it forms water. All right, so then that basically gets us to the ultimate result of breaking down glucose. You want to get hydrogens, right? And if you take the hydrogens, you break them apart into electrons and protons. The electrons are brought to the electron transport chain, okay, and that's where you see all of these electron shuttles here that are bringing electrons to electron transport chain. Well, where they got these electrons is the hydrogens that were on the molecule of glucose. Now the protons then, those are the things that are right here. You got all these protons that are here, okay? These came from glucose. And then what's happening is that they're getting pumped out, and then they flow through the ATP synthase. Some of them eventually they will form water, and the water will leave the system. It's not like you have these protons continually going back and around and around and around. If that were the case, there'd be no need to have additional glucose molecules breaking down. But the fact is that hydrogens do combine with oxygen to form water. The water leaves, and so you have to keep adding more hydrogens into this system. That's done by consuming more and more glucose. Okay, so that's why we eat. All right, so here let's do a little bit of review and try and sort out kind of some of these complex things. I've got a whole bunch of questions here. Go ahead and pause the video, and then I will see if you can answer these, and then I will go through them. And with these questions, I really do recommend that you team up with somebody and you ask them these questions. And can they answer all of these questions to the thoroughness that I'm about to answer them? If they can, then you know they've got a good grasp on cellular respiration. If you ask somebody these questions and they do not answer them to as thorough degree as I am about to, then you know you've got some studying to do. Okay, so keep that in mind, and I strongly recommend you get together with somebody and you quiz each other on this stuff, because this can be hard and easy. Can be easy to get confused. So go ahead and pause the video and kind of work through these. All right, here are the answers. This first one's pretty simple. Oxidation is the loss of electrons, okay? Just remember Leo the lion, loss in electrons is oxidation, and reduction is then the gain in electrons. The general equation for the process of aerobic cellular respiration looks like this. You take some glucose, 1 molecule of it. For every 1 molecule of glucose, it requires 6 molecules of oxygen. Together then, the products then are going to be 6 molecules of carbon dioxide, 6 molecules of water, and also a whole bunch of ATP, about 36 molecules of ATP. All right, so this is kind of a complex equation, and there's a lot of stuff here. Okay, there's a whole bunch of atoms in glucose. There's a whole bunch of atoms here in all of these oxygens. There's a whole bunch of atoms then CO2 and water, H2O, and ATP. It's hard to sometimes keep everything straight, but I'll give you a secret tip here. A lot of this stuff is just kind of stuff. It's pawns in the whole process. And so basically what's happening is that the long and the short of it is that the carbons of glucose and the oxygens of glucose, okay, together with the oxygen that we breathe, this stuff makes CO2 and water, okay? And it's the hydrogens that are really used to make the ATP, okay? So these are kind of, I think in my mind, the more important things to keep track of, because the rest of this stuff is just stuff coming and going along the ride of the process, along the journey. They're just kind of steps in the intermediate steps in the thing. But the thing that's actually making ATP is really the hydrogens, at least from how I see it. Okay, so here's kind of what's really going on. You are taking the hydrogens. Again, I've already kind of done this a few times. You're breaking them into 12 electrons and 12 protons. The electrons get delivered to the electron transport chain. The way that they are delivered is they ride on these molecules called NADH or perhaps FADH2, okay? But generally NADH. That's how they're getting the electron transport chain. The purpose of them is to pump protons up concentration gradient, okay? To pump hydrogen ions into the intermembrane space and thereby establish a strong concentration gradient where there's a lot of protons in the intermembrane space and not as many in the matrix. Then, so the electrons are actually, the ETC, the electron transport chain, is actually pumping these protons that originally came from the hydrogen. And then the job of the protons is pretty simple. Okay, all these do is they just flow down the concentration gradient. And by doing so, this is through the ATP synthase, carrier protein, and that is going to take ADP. It's going to phosphorylate it, add a phosphate to it to make ATP. Okay, that's kind of the whole long process. So then when we go back here, this kind of keeping all of this in mind answers all of these other questions. What role does the electron transport play in aerobic cellular respiration? Well, it is going to pump hydrogen ions up their concentration gradient. That is to say, it's going to establish a proton gradient, was probably the more succinct way to say it. Meaning there are lots of protons in the intermembrane space. Okay, from where do the electrons supplying the electron transport chain come? How do they get there? Well, they come from glucose ultimately, specifically the hydrogens of glucose, so the H12 of glucose, and they get there by these things, NADH and FADH2, the electron shuttles. And don't let these big scary molecules confuse you. When you see this, just know that this thing is just an electron shuttle that's carrying 2 electrons on it. And so is this, 2 electrons. Okay. What molecule is the final electron acceptor for electron transport chain? That's oxygen. So remember, we've got electrons and also protons merging with oxygen, and this forms water. Okay. And then chemiosmosis. Oh, this is one that I forgot to talk about. The process by which protons flow down the ATP synthase is referred to as chemiosmosis. So I should probably go back and show you this. This process right here, this is the ATP synthase. When hydrogen ions are flowing down through this, that process is given the name chemiosmosis. And it's in part, I think, because as these hydrogen, osmosis usually refers to water, and chemi probably refers to chemical or something. And I don't know exactly why it's named this, but I do know that these hydrogens will flow down through, and then they'll be available to then meet up with oxygen and form water. So I wonder if that's kind of what the name is hinting at. But it generally refers to the flowing of hydrogens through the ATP synthase. Okay, so that's oxidative phosphorylation. So we've kind of talked about this whole process. And now we are going to put a check mark there, and now we're going to turn our attention back to, we're going to go back to the beginning here to get our bearings on how the first steps of this whole aerobic respiration proceed. So this is kind of a figure that shows the step-by-step process here. We start with glucose. And glucose gets broken down, as we said, into 2 pyruvate molecules that are each 3 carbon molecules. Glucose is a 6 carbon molecule. Okay. And what's happening is you're also making NADH, and you're also making 4 hydrogen ions. So 2 NADH is the same thing as 4 electrons. So you've taken glucose, you've stripped off 4 electrons, you've stripped off 4 protons, and then the result is that you ended up with some pyruvate, and then you got a little water as a byproduct, and you've also made some ATP. Now, keep in mind the ATP that's made here, this is not the ATP that's made. It's the same as the ATP made inside the mitochondria. This whole process here, glucose getting converted to pyruvate, this is called glycolysis, and this happens in the cytoplasm, not the mitochondria. And we said that oxidative phosphorylation occurs in the mitochondria. So certainly then the ATP that are made in this step could not be made by oxidative phosphorylation, meaning the flowing of hydrogen ions through an ATP synthase. So these are actually made in a more direct fashion. Basically, some energy from enzymes and chemical reactions happening in the cytoplasm basically just take ADP, they add on to it some phosphate, and you get ATP. So it's the same kind of thing that you get in oxidative phosphorylation. It's just the thing that's supplying the energy here to make these two bind together is not an ATP synthase per se, as it is just the kind of the energy resulted from different chemical reactions in the cytoplasm. When this type of phosphorylation is occurring, we call it not oxidative phosphorylation, but we call it substrate level phosphorylation. And this is generally how bacteria can make energy. Bacteria do not have mitochondria, right? They're prokaryotes. And so they still have to make energy, and this is kind of one of the ways they do it. They mostly just through this whole glycolysis event. And this works. It does make energy, but it's not nearly as efficient or robust of a process for making ATP as oxidative phosphorylation. Okay, just keep that in mind. All right, pyruvate then is going to have so it's going to have 2 electrons stripped off of it. So that's, 4 more electrons stripped off of it. So that's where we get these NADH molecules, and then also some carbon stripped off to it. So that's why we have a little CO2 as a byproduct now, and that makes sense because I had 2 3 carbon molecules, and now I only have 2 carbon molecules. So this is 6 carbons total. This is only 4 carbons total. Well, what happened to those other 2 carbons? well, they're right here. You breathed them out as CO2. Okay. This whole process is a little bit of a smaller step in the whole game here, but this is called the oxidation of pyruvate. Because you're taking pyruvate, you're turning it into some substance and that has actually had 4 electrons removed from it, right? Because you have acetyl-CoA now plus these 4 electrons. So these were stripped off of pyruvate. When you have a loss in electrons, we call it oxidation, right? So this is the oxidation of pyruvate. The next step then is to take acetyl-CoA and basically just strip every single last thing off of it. So you take acetyl-CoA and it gets totally broken down, and now there are 12 electrons that have been pulled off of acetyl-CoA. Actually, scratch that. A total of 16 electrons, because there's 2 on this molecule and there's 12 together in these. So you've taken acetyl-CoA, stripped 16 electrons off of it, 6 hydrogens off of it, 4 carbons off of it, and 8 oxygens off of it. So basically all the remaining atoms that were originally here are now completely dismantled. Okay, this process is a pretty extensive process known as the citric acid cycle, and this occurs inside the matrix of the mitochondria, so the very innermost part. The citric acid cycle is really complex. It looks something like this, okay, and if this was a biochemistry class, you might have to actually know all of these steps, but in this class I'm not going to ask you to know these. But basically acetyl-CoA, which looks something like this, turns into citrate, then isocitrate, then alpha-ketoglutarate, then succinyl-CoA, then succinate, then fumarate, malate, and then oxaloacetate. Very complex process, and certainly don't worry about knowing it. But I do like to show you how complex it is. Okay, so that whole citric acid cycle, what happens is that this goes in and it kind of goes through a couple of turns before it's completely dismantled. And this whole biochemical process here is stripping, slow and sure, all of the atoms off of acetyl-CoA, so that you're then left with, you know, the electrons in these electron shuttles and then the hydrogens, and then you breathe out this stuff. So now we've got these electrons here, which have accumulated in the matrix of the mitochondria. Well, then the next step is going to be, you know, so let's say I've got, draw a picture of a mitochondria here. Here's the outer membrane, and then here's the inner membrane. So in the matrix is where the citric acid cycle is coming. So this is where everything's being completely dismantled. So you're getting a whole bunch of hydrogen ions accumulating in the matrix. And then in that inner membrane is the ATP synthase proteins, okay? And they work by hydrogen ions flowing down this way through it. Okay, so somehow these hydrogen ions have to get pumped up into the inner membrane space. The thing that pumps them up is the electron transport chain. Okay, so that's the little series of proteins that are found along this inner membrane as well. Okay, so that's going to pump the hydrogen ions into the intermembrane space. The electrons that supply that electron transport chain come from all different steps of this whole process. So anywhere you see these molecules, that means that they're bearing electrons, and where they actually end up going is the electron transport chain, so that all of these 6 hydrogens, which are in the matrix, can get pumped out. They accumulate in the intermembrane space, and then they flow down the ATP synthase. They turn it. They provide the energy to take ADP, add it to inorganic phosphate to make ATP. Now that whole process is known as oxidative phosphorylation, and here's where you see the difference in the efficiency here. 32 ATP are made in this. You know, you had compare that to 2 that are made here. Now this is the step that requires oxygen, because remember, when you had the electron transport chain, those electrons that are whizzing through it, they ultimately land on oxygen. So this is the one that requires oxygen, but if you do have oxygen around, then you're afforded to undergo oxidative phosphorylation, which is going to give you a lot of bang for your buck. You're going to get lots of ATP. It's super efficient. If there is no oxygen around, then it kind of ends right here. And you kind of have to settle with your 2 ATP. There's different types of muscle in your body that utilize these different types. Like, for example, marathon runners, they've got a lot of mitochondria and they're well suited to do this process, oxidative phosphorylation. It's very efficient, but it does take quite a bit of time to make ATP, as you can imagine. Taking a glucose and breaking it down to pyruvate and then breaking it down to acetyl-CoA and then breaking that down in the citric acid cycle, which is heinously complex, and then somehow getting that to flow down the electron transport chain and then the ATP synthase. It's a super long process, so it takes a long time. Lots of great energy made from it, but it takes forever. But that would be like a marathon runner. His muscles would have a lot of mitochondria well suited to that. Contrast that to like a bodybuilder's muscles. Bodybuilder's muscles are going to use a lot of this to make energy, and it's really, really quick energy. And so it's kind of an interesting tidbit there. All right, here's just kind of a quick recap of this whole process, just so you can see it again. Here is a cell. Inside the cell, this is the nucleus. All these little things that you see here are mitochondria. And for the simplicity of the figure, I've kind of omitted all of the other organelles in the cell. Basically, we have, again, glycolysis, the first step. And what happens here is that glucose is converted to pyruvate, and then you also end up with some some electrons and also some hydrogens as well. So here's my molecule of glucose. It's in the cytoplasm now, inside the middle of the cell. It gets broken down into, it's a 6-carbon molecule, and now each of these are 3-carbon molecules. And then you make a little ATP. There's some hydrogens and some electrons made. Then what happens is that these things are going to find their way into the matrix of the mitochondria. Keep in mind, these molecules that I've drawn here, they're not even close to scale. These are like a gazillion times bigger than they would actually be in real life, but I'm just drawing them so you can see them. Right, so these things are going to migrate their way into the matrix of the mitochondria. Along the way, there is going to be the oxidation of pyruvate. So now it's going to be forming this stuff called acetyl-CoA by the time it reaches the matrix. So now the molecules that we're dealing with here are acetyl-CoA. And then acetyl-CoA is going to, in the citric acid cycle, that super long complex cycle, it's going to be totally stripped down into a whole bunch of stuff. So it's broken down into a bunch of hydrogens, a bunch of electrons. These electrons are going to be flowing along the electron transport chain. When they do that, they trick hydrogen into popping up into the intermembrane space. And then the last part is that there's a oxidative phosphorylation, is that there's a ATP synthase protein in that membrane. And all of the hydrogens that have accumulated here, all of the protons, I guess I should say, that have accumulated here can now flow back down through. And then in the process, all of these electrons, they eventually, after they've run their course through the electron transport chain, they find their way to oxygen to make water. And that's ultimately what all of these that are lying around end up doing too, is they combine with the electrons again to make water. All right, here's a review question 2. Where does glycolysis occur? What are the main outcomes of glycolysis? Where does the citric acid cycle occur? And what are the main outcomes of the citric acid cycle? Go ahead and pause the video and work on these. All right, the answers. Glycolysis, cytoplasm. The main outcomes of glycolysis, well, you take glucose, you break it down to pyruvate, and in the process you make a few electrons, and you also make some ATP, okay, and a little bit of water, and also some hydrogens, ions. The citric acid cycle, and the big thing here to keep track of is that the glucose goes into pyruvate. I'm not too particular that you know all of the how many hydrogens and electrons and everything, but it is good to see that the electrons and hydrogens are being made because they're important for oxidative phosphorylation. Okay, the citric acid cycle, this is occurring not in the cytoplasm, this is deep inside the mitochondria now, the matrix of the mitochondria. And basically what's happening here is acetyl-CoA is completely being dismantled into electrons and protons. And then, you know, the carbon and oxygen kind of escapes as CO2. Then those electrons and protons, again, for the umpteenth time, protons get put into the intermembrane space. Electrons go to the electron transport chain, where they actually are the things that are responsible for making the protons go into the intermembrane space. All right, there you have it. The first kind of run through of aerobic cellular respiration.